All right, hello everyone. It's great to meet you here in this room. We're going to be chatting about some of the work being done here at Penn State on ticks and tick-borne diseases. We're going to get right to it, but I am going to warn you that this will take us right up to the end of our 15 minutes of time today, so just kind of hang on to your hats as we breeze through. It's going to be a lot of information, but we do have a Q&A session at the end of these rotations, as you know, so if you have any questions, just write them down and I'll answer them as soon as I can. So I probably don't have to do too much of an introduction for you all on the relevance of ticks and tick-borne disease in Pennsylvania. So we're just going to start with a 30,000-foot overview here and the release of the CDC report in 2018 that stated there has been a tripling of vector-borne disease cases in the country in the past 13 years, and 75% of those cases are actually tick-borne diseases. Now in Pennsylvania, we've been number one in cases of Lyme disease for many years with over 7,900 cases in 2018. But because not all cases are diagnosed and instead are treated based on clinical signs, the CDC estimates a 10 times multiplier. So we're actually looking at more like 79,000 cases in the Commonwealth annually. So hunters and hikers and campers and other recreational outdoors people are all at risk for tick encounters. Not only do we have human cases, but we also have dog cases of Lyme disease as well, which is of course a concern to pet and working dog owners, and to horses, which are a $3.3 billion industry in Pennsylvania. Now, there's more to the story of ticks and tick-borne diseases than Lyme disease, however. Along with Lyme disease, we've seen anaplasmosis cases rise in dogs and horses, and the invasion of the Asian longhorn tick, which can infest horses and may transmit pathogens that are fatal to cattle. So Lyme disease and other tick-borne disease cycles are extremely complicated, as you can probably see from this extremely complicated figure, but most ticks haven't been studied to the same extent as other vectors like mosquitoes. In fact, it wasn't until the early 1980s that Lyme disease was recognized as a real concern. And it was also during this time that there was a real increase in the use of omics, if you will, or laboratory-based genetic research to study organisms. Now, while these are powerful tools to study the physiology of ticks, the challenge with using this very basic science is you fail to understand the biology of the organism you're studying in the habitat where it actually encounters people and animals. And this is where we have some of the largest gaps in our ability to control ticks. We're missing critical information on tick ecology that would allow us to find ways to interrupt that ecology for control. So while tick-borne diseases are complicated, a better understanding of tick ecology and behavior is actually a focus of much of the research being done here at Penn State. And today we're going to talk about just a few specific projects that are currently in progress, including testing available tick control tools, achieving a better understanding of host use by ticks over the landscape, understanding how ticks locate and select their hosts to bite, and then, of course, engaging the public to increase knowledge of ticks and tick-borne diseases through the development of the Vector-Borne disease team here at Penn State Extension. So let's get started talking about tick control. So tick tubes are a user-friendly tick control tool. These cardboard tubes are commercially available, they're biodegradable, and they consist of cotton impregnated with permethrin. Now, the mode of action is very simple. So mice, which are common hosts for immature ticks and the main reservoir for the pathogen that causes Lyme disease, are the main target for this treatment. And the idea is that mice will interact with the tubes and remove the cotton as nesting material, which treats themselves and their nestmates for ticks, with these very low doses of permethrin that are found in the cotton. Now you can see in this video the top of a mouse head pulling cotton from the tube, and then it jumps on the log to take it back to its nest. One of the challenges with this product, however, is that while interactions with the tubes have been tested in the lab, an understanding of how the treated material is used in the field by mice during critical times of the year when the immature ticks are present has not been evaluated, which may lead to unsatisfactory control. So we've partnered with Thermacell, which is one of the tick tube suppliers, to study how mice interact with the product with the goal of improving the use of tick tubes as a tick control method. So last year we did a small field study to evaluate the tick tubes on a local scale to determine if they actually remove ticks from mice, and a copy of the study is in the resources that were sent to you. And I had two undergrad students lead the study, Jessica Brown and Taylor Miller, who were lead authors on the paper. And they demonstrated quite nicely that the addition of the tick tubes reduced ticks on mice from about 4 to 7 ticks per mouse in our control groups, which are the top numbers in each red box, to 0 in the treatment groups in a matter of 4 weeks. Now this year, we're evaluating mouse use of the tubes using trail cameras, as you can see here, and testing some improvements to the product to increase the use of the cotton by mice by adding either different cotton sizes or by using different odors. So we know that mice primarily forage by using odors, so we've selected highly volatile odors, including peanut butter, safflower oil, or vanilla, to increase mouse visitations and hopefully then increase the use of the cotton. So what you'll see here is Kylie Green and Chloe Robertson resetting both trail cameras to video the mice and also the different tube treatments during our August trial to evaluate these improvements. And in the next video, you can see a mouse visiting and interacting with the tick tube material, again, treating itself for ticks. Now, the more material that these mice pick up and use, the more protected they'll be from ticks, hopefully breaking both the tick life cycle by killing the ticks and then also interrupting the tick-borne disease cycle. So tick tubes, like I said, are a very consumer-friendly tick control tool, and if we can improve how they work, we can help the citizens of the Commonwealth reduce tick bite risk. And so far, we've seen some pretty clear differences in how mice use these tubes with these improvements. If you look on the figure on your left, you can see that the small cotton seems to be used more frequently than the other sizes, including the commercially available Thermacell tubes. And on the right, you can see peanut butter and safflower oil odors added to the cotton increases use as well over the commercially available tubes and vanilla. And we finished this research in October. So we're also interested in how ticks use their hosts across the landscape and what hosts are used for feeding. So we've incorporated those questions into most of the projects that we get started. And the idea is if we can better understand what animals are used by ticks and where on the body the ticks are, we can target those animals or those areas for better control. So we're currently evaluating ticks on white-tailed deer and deer use of suburban landscapes as it relates to tick density and tick control methods. And we're doing this in collaboration with the University of Maryland, the Connecticut Experiment Station, the US Army, the CDC, and the USDA. And this project required live trapping deer in park settings and fitting them with GPS collars to track their movements over the past several years. Now, one of the wonderful parts of this project was being able to use my wildlife experience to actually execute the deer trapping component. So I'm going to take all of you now on a brief tour of what live trapping deer looks like. So initially, we have a team waiting out of sight in a vehicle with most of the equipment. I'm actually up on a hill in a deer blind waiting to drop the net on the deer. And once we drop the net on the deer, we all rush to the site because we want to subdue those animals to reduce stress and prevent injury. So we're all running down to the net. We put a blind on the deer, which again helps with stress and keeps the animals from jumping around. Once we subdue them, we can anesthetize them. So you can see me here in the camo jacket anesthetizing this deer. And again, we do this so we can work them up without stress. And then we do the workup, which includes monitoring vital signs, respiration, heart rate, that's a pulse oximeter. We fit them with a GPS collar. You can see here, all deer have different neck sizes. We put in an ear tag. Now this is the same kind of ear tag you'd see in cattle, so we can visually identify them. And then we can check for ticks. So we look for ticks around the ears and on the head, but also the other body regions between their legs, on their rump, on their sides. And we age the deer so we can get an idea of how ticks use different deer age classes. And once we're done, we can actually reverse the action of the anesthetic with its reversal agent. A deer will pop up in about 10 minutes and will be released. Now, this particular deer was actually too small for a GPS collar, but we were able to check it for ticks. And what we found is that deer use very targeted core areas in suburban areas, which may affect tick control. And we also found some pretty distinct tick locations on deer from different states. So in this figure, you can see the different colors that indicate tick body region dominance by state. And you can see that there are some pretty extreme differences in where ticks are located on deer, which may influence tick management on deer. So we also have a project in collaboration with the Pennsylvania Game Commission looking at tick associations with black bears and black bear movements across the landscape. So I have a graduate student, Hannah Tiffin, who you can see in this clip, who's working up a sow that has evaluated tick use of black bears and is working to develop a model for tick abundance on bears, the pathogens found in those ticks, and how bears move those ticks across the landscape. And one of the benefits of this project is being able to go out every winter on den checks to check GPS collars, but also to get to see some baby bear cubs. Now with the bears, Hannah's found that ticks attach to different body regions differently by month, which corresponds to the various life stages of the ticks. But also, and interestingly, that quite a few ticks are actually found on black bears. So this animal may play a significant role in moving these ticks across the landscape, which is a role that we've previously attributed primarily to white-tailed deer. So we've also evaluated smaller animal use by ticks. As I mentioned, mice are the primary reservoir for the pathogen that causes Lyme disease, but it's been assumed that black-legged ticks use whatever host they find first. So we've been trapping rodents in Pennsylvania for 3 years now, evaluating pathogens found in those rodents and the ticks that we remove from them. Now we find ticks on all areas on the body of the mouse. As you can see here, we even search in between their little toes, and we can recover anywhere from 0 ticks to over 30 ticks on a single mouse. Now, however, we have observed in the field that we can find an average of 7 ticks on a mouse, but less than 1 on this critter, which is the southern red-backed vole. Now, these two animals are in the same habitat and are trapped at the same time. So the question is, what is causing this difference? Now, if we could identify if ticks are choosing mice over voles and why, we may be able to use that information to develop novel control methods. So this leads us to the next section, which is a newer project in my lab, where we are looking at the fundamentals of tick host selection and what causes ticks to select one host over another. Now we're looking at both of these questions through the use of what's called an olfactometer. In this case, a YouTube olfactometer, which you can see in this video that has a few ticks in it. So ticks are responding to air that has been mixed with either mouse or vole odors. And you can see ticks here that have climbed up to the end of this tube, having selected a side of either mouse or vole odor. And then you'll be able to see the whole system here where purified and humidified air flows through to the ticks. And using this system, we can gauge preferences or even repellent behavior if the ticks don't like a particular odor. So we'll also be evaluating the behavior of mice and voles with ticks with the use of EthoVision software starting this fall. So EthoVision is a behavior tracking software, as you can see here. And we'll be using the software to see if we can find differences in how these animals behave when they're infested with ticks. And finally, and probably most importantly, research has shown that the best way to reduce tick bites on an individual level is education on the topic. So in response to the CDC report and to fill the gap in extension education, Penn State Extension has developed the Vector-Borne disease team, which is focused on tick prevention and control. To start this team, we distributed a needs survey, which received over 3,000 responses from citizens of the Commonwealth, which has directed our educational programming. And our approach to this topic is unique in the area, as we are not only the only extension service with a vector-borne disease focus, but we also have programming for not just people, but for animal protection as well. So Jessica Brown, who I mentioned earlier, and Dr. Karen Poe, who's a postdoc scholar in my lab, who you can see in this video, are two of the team members who have initiated the Tick Safe Pennsylvania program as part of the vector-borne disease team. Jessica was a major in wildlife science here at Penn State and remarked that she was never trained or even warned about tick risk when learning in the field during her program. So clearly there's a training gap. So initially, Jessica and Karen are evaluating tick encounter risk in some of the high use areas on campus and will be eventually producing training materials for the university that can be packaged as a toolkit for other institutions to train faculty and students on being tick safe. Now, we're currently developing educational content, including Learn Now videos and webinars and articles and other programming that'll be part of the Tick Safe Pennsylvania campaign. And you can find some of these resources and the materials that were sent to you as part of this tour. And I also invite you to watch some of our webinars that were given live through the summer and were recorded and are now on the Extension website. Okay, so with that, I'm not sure if we have much time. This is a whirlwind tour of the ticky work done at Penn State, but that's what I have.